Hey everybody. Yeah, still sick. Got diagnosed with influenza A. Um, and I think we talked about vaccines earlier and how um, they have to they have to produce the vaccine and guess. I mean, obviously an educated guess, but they have to guess how the virus will mutate um, in the few months before cold and flu season. So the vaccine becomes available around August, September. However, the season for flu isn't really until January or December through February, a few months later. So what I'm trying to get at is sometimes the virus, sometimes the vaccine is very good. And I did get the vaccine, by the way. And sometimes the vaccine kind of misses because you don't know exactly how that vaccine you don't know exactly how the virus is going to evolve and how it's going to change. And so I always tend to put things in terms of biology. And I find this one interesting because one of the things that this particular strain is doing is it's making me cough a lot. And if you've ever watched the YouTube video, and maybe I'll play it for you when I get in class, there's a YouTube video of somebody sneezing, and the amount of stuff that comes out of your mouth is really amazing. And I am still coughing a lot. I think I could come in today, but because of the fact that I'm coughing so much, I don't want to be spraying that all over you and getting you sick. So, anyway, long story short, again, I'm going to take today off and hopefully I'll be back on Thursday. With that said, I'm going to skip over the part of where we talk about meiosis because this is a fairly straightforward unit. The only thing that's a little bit sometimes harder to understand is the meiosis bit. And so I want to make sure that I'm there for that part so that you can ask questions if you have them, because I'm sure some of you are gonna be like, I'm not sure what's going on, and you want to ask questions and you can't do that with a video. So we're gonna go to slide 54, which is chapter 14, and that's genetics. So I know a lot of you have had genetics in the past, and hopefully that will, this will be kind of a respite from some of the other stuff we do. And then after that, we will go into we'll go back to the meiosis part when I'm back in class. Okay, so Gregor Mendel, um, was a monk who studied botany, and he loved working with garden peas. And garden peas produce, at least the varieties that he was working with, produced all sorts of different characteristics. So they had a lot of different varieties, a lot of different things that he could study. He could study the color of the flower. He could study how high. Some of them were dwarf, so they didn't get very high. Some of them had different types of um, what do I want to say? The peas had different shapes, the actual peas themselves. So anyway, that's what he studied, and he was able to find some things that other people were not able to find, and there were a few reasons for that. So other advantages to using these garden peas, it doesn't take very long for them to produce um, seeds. So to go through an entire generation doesn't take very long. So just to give you a little more background, I don't know how much you know about plants, but we always think of, or I think of anyway, plants as flowering plants, and pretty flowers. But during that flowering process, the reason for that flower is to reproduce. And so after the flower has hopefully been fertilized or done its fertilizing, and we'll talk about that in a second, then you will produce seeds. And then that's what I mean by a short generation time. So then those seeds will be produced after the flower's gone, and then you can take those seeds and you can plant it in the ground again. Um, the other advantages of using peas is that you produce a large number of offspring, and we'll get to that one too. It's kind of cray what Mendel did to get all of his data. And then mating can be controlled because many flowering plants are what we call self-pollinating. That means that they have both male and female parts. So self-pollinate means that they have both sperm and they have egg. or they can be cross pollinated. So you can take the male parts and you can use those male parts and fertilize a different plant with the sperm of one plant. So one plant can fertilize another one. So that's cross pollination. Um, Here's a picture of what he did. So he took flowers and he cut out these things called the stamens. And in the stamens, he actually he cut out those stamens from in this case, the purple plant here. So he took these and he cut them out so that the plant now only had female reproductive organs rather than both male and female. So it could no longer self pollinate. And then he took a different flower, and in this case, it was white. And he took a little paintbrush, and he took the pollen off the stamen of the white flower. So this is the male part. And he brushed the tip of that brush against the stamens of the white flower, and then put them on the carpel. And you don't need to remember these names. The carpel is the female part of the plant, and you put the sperm from the white flower onto the egg of the purple flower. In other words, and that's what we call cross pollination. So you're taking the pollen or the sperm, pollen is sperm, from one plant, and you're using that on a different plant. And so he took it from the white one and put it on the purple one to see what he would get. And when he did that, oh, sorry, let me before we get to that, before we get to the results, let's talk about the fact that he also started with varieties that were always true breeding. What does that mean? It means that if you just let the flowers um, self pollinate, if they just self pollinate, you're always going to get the same outcome. So if you had a purple flower and you allowed those um, to just self pollinate, you would always get purple flowers. If it was a white flower, you'd always get white flowers. If it was round seeds, you would always get round seeds. If it was a dwarf plant, you'd always get a dwarf plant. Okay, so true breeding, always getting the same traits. And in that last figure, that's what he was using. He was using purple plants that were true breeding, and he was using white plants that were true breeding. So what did he get? The experiment that I just showed you in that picture, that's called hybridization, where you take two true breeding varieties and you cross them. The true breeding parents are called the P generation or parental generation. And the hybrid generation, where you cross the white and the purple, are called the F1 generation. After he did that, after he took the purple and white flowers and he crossed them, then he let them, rather than cross pollinate again, in the second part of the experiment, that's called the F2 generation, he allowed them to just pollinate on their own. Okay, so no cross pollination in that. I know it says cross pollination with others, but in the original one, he didn't do any cross pollination. He just did self pollination in the F2 generation. Okay, now let's get to what we got. When Mendel crossed the contrasting true breeding white and purple flowers, all the F1 hybrids were purple. Okay, so he crossed white and he crossed purple flowers that always produced white or purple. And in the end, all of them were purple. Okay, that's okay. What happened to the white ones? So then Mendel took those F1 hybrids and he crossed them. Okay, he crossed an F1 with an F1 and that were purple. Okay, they were purple in that first generation. But in the second generation, when he allowed them to cross, he found that they were in the F2 generation. Oh, sorry about that. He found that now all of a sudden he had white flowers again. He had about, on average, 3 purple flowers to every 1 white flower. So his ratio was 3 purple to 1 white. Okay, so first generation, F1 generation, all purple. And I think that's actually just shown on the next page. Let me just go there. So purple and white flowers, true breeding. They were all purple in the F1 generation. So this is F1. But then when he crossed them again, um, now he got 3/4 of them being purple and 1/4 being white. So what does that mean? Well, Mendel reasoned that the purple flower was the only flower in the first generation that was affecting the outcome of the color. And so, because of that, he called the purple flower, the purple color, the dominant color. Called it the dominant color or dominant trait, while he called the white one the recessive one. Okay. Um, the fact that he got white flowers in the F2 generation, that it just reappeared and bada boom, bada bing, now it was white again, suggested to him that the white flower hadn't been like diluted. So it wasn't like the purple was so overwhelming that it just diluted it, and the white flower was gone forever because it came back. We saw it in subsequent generations. So it suggested it was still there. It was just hidden for some reason. Mendel observed the same pattern in 6 other pea plant characteristics that each were represented by 2 traits. So you either had purple or white, or I'm going to show you the chart in a minute. You had round or wrinkled seeds. You had dwarf or tall plants. So in all those cases, he found the same deal where one trait was hidden in that F1 generation, and then in the next generation, they suddenly reappeared. Mendel knew that something from that purple color or the seed shape, texture, was being passed down. And he called that a heritable factor. He didn't know what a heritable factor was. This was in the 1800s. And they had almost nothing in terms, they might have had like very basic microscopes, but there's no way they could look at genes. So we know now that what he's calling a heritable factor is actually a gene. But at that point in time, they had no idea. So he was pretty clever in figuring this all out. Um, so I know this is really small, and you may not be able to see it really good, but I'm gonna read you off some of the things. So in each one of these cases, when he counted the F2 generation, so the second cross, he found a pretty close. And remember, we're talking biology. We're talking there's some probability in here too. So he consistently found around a ratio of 3-1. 3 for one trait and 3/4 having one trait, so 75%. And then the other 1/4 or 25% showing the other one. So, for example, um, in addition to purple and white, he looked at seed color. So he looked, were they yellow seeds or were they green seeds? He looked at the shape of the seed. He looked at the pod color, all these different traits. Here's the stem length. I told you dwarf versus tall. But what I wanted to point out was this is real science back in the 1800s. Here's how many seeds he counted. You add that up, that's over 8,000 seeds. And if you add the seed shape, that's close to 8,000 as well. Okay, so he did a lot of counting to do this. So there's a lot of, dirty work, meticulous work that had to be done. And he did a very good job of doing it because he came up with what we know now is true in terms of inheritance of traits. And we now call it Mendelian genetics. So Mendel developed a hypothesis to explain the 3-1 inheritance that he saw in the F2 offspring. And so he had 4 related concepts that made up that model. And these concepts can be related to what we know now about genes and chromosomes. Now, again, he had no idea what a chromosome was. He had no idea what a gene was at this point. So he was pretty ahead of himself, ahead of his time. Okay, first idea is that there are alternative versions of this. And again, he called it heritable characteristics. Okay, but we know it now as there's different variations of a gene. Okay. And that accounts for the variation that you see in inherited characteristics. For example, the gene for color in flower color in pea plants exists in two versions. You can either have purple or you could have white. Those different versions are what we call alleles. And I don't know if you've been taught that word or not, but alleles are just different variations of a gene. Okay, we could also talk about it in terms of eye color. So with eye color, it's not as straightforward, and we're not going to talk about that right now because it's more than we can do. It's more than we know right now at this point in the lecture. But we could do eye color, and eye color would have blue gene, hazel gene, green gene, and brown gene. Okay, there's more than just those two alleles. But for the peas that he was looking at in flower color, there were just two versions. You either had purple or you had white. So again, he didn't know this, but we know now that what that means is that an allele has a different code. It has a different code of A, C, T, and G. Okay, so we talked about DNA and we talked about how that code gives us a particular gene. So in each of these cases, there is an allele that codes. One codes for purple and one codes for white. And they're both located on the same, I'm making this up. I'm going to say chromosome 1. This is on chromosome 1 as well. And they're in exactly the same spot on chromosome 1. So that's the gene locus, right, or location. But the sequence is different. And we know now that that sequence it starts with a C, and if you're purple, and it starts with an A if you are white. Okay, so you produce different things depending on what that allele version is. So that was um, his first idea. The second one was that an organism inherits two alleles. They get one from each parent, right? We talked about chromosome 1. You get one chromosome from one parent, and you get one chromosome 1 from the other parent. Okay, so um, each parent's going to donate, or not donate, but it's going to give one allele for that particular characteristic. If those two alleles are the same, then we call those true breeding. But if they are different, we call them hybrids. Okay, and you're going to see in a minute that that is also referring to the idea of having dominant versus recessive. Okay, so his third concept was the idea that if the two alleles at a locus different, then there's going to be what we call a dominant allele that shows itself. Dang it. There we go. A dominant allele that will show itself, and a recessive allele that will not. Okay. In flower color, the F1 plants had purple flowers because the allele for that trait, the purple trait, is dominant. So I could say that it'd be big P. Okay. That's the dominant purple allele. And then the other one is a small lowercase p, which in this case is we always use the letter, well, not always, but often the convention is really to use the dominant allele, use the first letter of that particular trait. So we use P for purple. But then the recessive one is going to have the lowercase. So I have a lowercase p here rather than having, we don't use a W in this case. Okay, we don't use a different letter. We use the same letter, we just use a lowercase. And so I've got a dominant and I have a recessive. But it's only going to be the dominant that shows itself. It's only going to be the purple that shows itself. Fourth law is, or fourth concept is the idea of what we call law of segregation. And this one will be a little easier to understand when we've gone through meiosis, but the idea is that we do know from our previous lecture that during meiosis we go from 46 chromosomes to down to 23. Okay, so even though we have two copies of a gene that we got from our parents, when we pass down our allele, we only pass down one of the alleles. We don't pass down two of them. So we get one allele from each parent. Excuse me. Um, so we'll come back to that one a little bit later because I think it'll make more sense after we've done a little bit more with meiosis. Okay. The model that he came up with accounts for the 3-to-1 ratio that we see in the F2 generation. And we can actually do some calculating or estimating of what will actually happen by using what's called a Punnett square. So in a Punnett square, what we can do is we can use that technique to basically predict what the offspring will look like. And so there's an example of that here. I had purple flowers, and now that we know that there's these allele things, we can start talking about that. So these purple flowers, we know they're true breeding, which means that their alleles have to be big P, big P. And we know that the white flowers, their alleles have to be small p, because they always produce white. Well, when you do that cross, you end up with the only allele that these white flowers can give is a small p, and the only allele that the purple ones can give is a big P. So that means that all of your first generation is going to be big P and small p. Okay. Now we can use something called a Punnett square. And a Punnett square is going to allow us to predict what the offspring will look like. So at the top, you're going to have if it makes a difference, and in this case it doesn't, but if it does make a difference, you're going to have the female at the top, and you're going to have the male is going to be on the side. Doesn't really matter in the case of flowers, because they have both parts. But I would say, okay, I'm going to take these purple flowers and I'm going to cross them with some more purple flowers from that first F1 generation. To make my Punnett square, I'm going to take one parent and I'm going to put him on the side here. And I'm going to take one parent and I'm going to put him on the top. I'm going to place one letter next to each row on the side. And I'm going to put a letter next to each column on the top. And then I'm going to take my letters and I'm going to combine them. So I'm going to get big P, big P. I'm going to get big P, small p. Notice how I always put the big P first, and because it's dominant, you put the recessive one after that. So I do the same thing down here, and then I do that. So if I look at this, I know that 1, 2, 3 of my offspring, or 3/4, will be purple. Unfortunately, I can't do white and white, so let's use black instead. And 1/4 of my offspring will be white. Okay? And that's a Punnett square. And we're going to use Punnett squares a lot in this unit. And your essay is actually going to be Punnett squares rather than an essay question. So just keep that in mind. I'm going to have you do some work on that after we're done talking. But the first thing I want to do is I want to talk about some vocabulary that when we do this, we need to understand, because we're going to use it a lot. And on the test, you might actually have these terms given to you. If an organism has two identical alleles, it's called homozygote. Homozygote. Or we say that they are homozygous for a particular trait. Okay. The other thing you can have is heterozygote or heterozygous. And that means that you have different letters. So for this homozygote, let me go back up here. That would be big P, big P. But it would also be or small p, small p. I really hate my Chromebook. It's so slow. Sorry about that. Um, whereas heterozygote is going to be this. Okay. Homozygotes are always going to be true breeding, right? Because they only have one letter, so they can only give one letter or one allele. Whereas heterozygotes are going to have two different alleles, so they could give a dominant trait, a dominant allele, or they could give a recessive allele. Let's see, we got some more vocab here. Yeah. Okay. So organisms' traits don't always reveal its genetic composition. So, in that F1 generation, we know that there was a white allele in there somewhere because they were um, big P, small p. They had that white allele, but that white allele wasn't showing that it was there. Okay. So we distinguish something between what a phenotype was, which is what it looks like. So it looks like purple. Hopefully I spelled that right. Not really. Oh, sad face. Okay. It looks like purple, and that is its phenotype. Okay. Phenotype is what they look like. It's their physical appearance. And then genotype is going to be the physical makeup. So in this case, we would either say that the genotype is big P, big P, small p, or we could also say that it is heterozygous. Okay, this is heterozygous. Um, because there's one big allele and there's one small allele. Hi, Kayani. How are you? My dog just came up to say hi. Yeah, I know it's time to eat. You could just wait a little bit, okay? Yeah, go away, please. Sorry. Okay. All right. So genotype, genetic makeup, the actual allele combinations, the P's, or heterozygote versus homozygote. Whereas the phenotype is going to be what they physically look like. Um, okay, so those purple ones, in that cross that Mendel did, some of his flowers were homozygous; they were true breeders. But some of those were heterozygous, so they were big P, small p. So even though they were purple, um, they had a different genotype. Okay, so genotypes and phenotypes don't have to be the same. Um, and that's the thing is, it's like, well, how do you know if it's big P, or how do you know if it's big P, small p? And just looking at it, obviously you don't. So you can do something that's called a test cross. And a test cross is nice because it allows you to see what's actually going on in a particular situation. It'll tell you, is the genotype, is it a homozygous dominant? Let me go back here one more time. Is it a homozygous dominant genotype in the case of this guy? Or is it heterozygous in the case of this guy? Okay. And so we do this thing called the test cross. An individual with a dominant phenotype could be either homozygous dominant or it could be heterozygous. So we can do what's called a test cross. And the way we do this is we test it with something that is homozygous recessive. So, like I said, you need to know these vocab words. So, if it's like, I don't remember what homozygous recessive is, make sure that you're up to speed with that pretty quickly because I'm going to use that a lot. I will tell you right now that homozygous dominant is that, heterozygous is this, and homozygous recessive is that. Okay, so what I'm saying is you're going to take whatever individual you're not sure of and you're going to cross it with something that is homozygous recessive. Okay, something that is a true breeding, but it shows the recessive trait. In this case, white. Okay, so here's the cross. They started off with their question. I don't know if this is purple or I don't know. I don't know if it's homozygous dominant or if it's heterozygous. I don't know which. I know it's purple. And they're going to cross it with something that is homozygous recessive. When you do that, you're going to get two different outcomes depending on what you started with. If you started with an individual or a flower in this case who is homozygous dominant, big P, then every one of the offspring should be purple. You shouldn't see any white. Okay. If you see any white whatsoever, and you really should see about 50-50 based on the Punnett square, then you know that you have a then you know you had a heterozygote that was purple. Okay, so rather than homozygous dominant, it had to be heterozygous purple. Oh wow. Okay, well, I guess you got to hear that whole coughing fit. Sorry about that. I tried to hit pause and evidently it didn't work. Let me hold on and do that again. Okay, that's better. And that, boys and girls, is why I'm staying home today rather than coming in and making you all sick. Okay, let's see where we're at time wise here. We're at 31 minutes. Okay, we're good for a while. So let's talk a little bit more about what we're doing. If I, there we go. Okay. Mendel derived the law of segregation by following a single character, allele, or sorry, gene. The F1 offspring produced in this cross is what we call a monohybrid cross. Because mono meaning you're just crossing two to produce a hybrid, mono, one. Across such heterozygous is called a monohybrid cross. Okay, that's really repetitive. Don't know if we really needed that. You can also do what's called a dihybrid cross, and di meaning two, and hybrid again, crossing two different characteristics. So having plants that are heterozygous for two traits. And that is where I'm going to stop because to explain this, I really do need to talk more about meiosis, and I can't do that right now, or I don't want to do that right now. So, anyway, what I'm going to do is I'm going to share with you the genetics PDF and have you start working on some of those. You'll be able to do some of the crosses. You won't be able to do all of them. As we go on in the material, you'll be able to complete more and more of them. So do what you can for right now. And then hopefully I will see you all on Thursday.